Hello everyone, my name is Matthew Back and I'm from Harper University in the UK. I was asked to give this keynote presentation by the conference organiser, Vincent Mitchell, and I'm very grateful for this invitation, and it's a great honour to be able to talk about this subject at the conference. The title of my talk is Crop Production Systems with Reduced Pesticide Inputs. The role of biofumigation in sustainable pest, weed and disease management. During this session, I hope to give you an overview of the work that has been done on biofumigation, but also talk about some of our own work that we've been doing at the university. There are a vast number of pests, weeds and diseases found worldwide, and these have a range of different effects on our crops. Ultimately, they accelerate crop losses whilst also increasing the variable costs to the farmer. The FAO estimates that we lose between 20 and 40% of our crops due to pests and diseases. The annual cost of diseases is estimated to be $220 billion, whilst invasive pests are estimated to cause $70 billion worth of damage. Added to this, we need to produce more food by 2050, with the estimate being around 25 to 100% more production needed. The movement away from traditional crop production systems where a diverse variety of crops are grown on a smaller scale to larger scale systems which are highly mechanized, where there are lower diversity in crops, so-called monoculture, has had a significant impact on epidemics of pests and pathogens. Additionally, the age-old problem of relying on a limited number of crop protection strategies such as varietal resistance or chemical controls also shapes the epidemics, where boom and bust comes into the equation. There are numerous examples to highlight the adaptation of pests, pathogens and weeds to the overuse of resistant varieties or pesticides. Just think about what's happened with potato late blight and how that pathogen has adapted to fungicides such as metalaxyl or fluazinam more recently. Another example would be yellow rust and the diversity of races and the selection of those races through the overuse of resistant varieties. This slide gives us another set of figures that were calculated in a recent study published in Nature Ecology and Evolution by Savary et al. And we can see the losses that are currently occurring in the 5 most grown crops around the world. We are all here at the conference to discuss alternative methods for managing pests and diseases, namely biocidal plants. And one of the reasons why we're all interested in this is because chemical control options have gradually diminished over the years. And the reason for that is that chemical control may have negative effects on the environment, such as the contamination of water. Some chemical controls have affected the ozone layer. There have been pesticides that have known detrimental effects on human health, and also the non-target effects of some of these chemical pesticides on pollinators, such as the neonicotinoids. And this slide really gives us a few examples of some of the loss of these chemical control options. And this is not a comprehensive list. This is just a set of examples that I've pulled out. Of course, one of the well-known losses is methyl bromide, very widely used soil fumigant for pests and pathogens, a very mobile gas that was phased out from 1998 to 2005 under the Montreal Protocol and the Clean Air Act. There have been others, such as 1,3-dichloropropene, banned in Europe in 2011. The seed treatment, Virum, for treating seed-borne pathogens was banned in 2019. The same year, the insecticide pymetrozine went. Glyphosate is gradually being phased out in various countries. We already mentioned the neonicotinoid ban because of its non-target effects on pollinators. In 2019, we lost chlorothalonil, which is a widely used fungicide protectant. And then recently in the EU, we lost the nematicide Vidate, which contains octamile. And I can tell you from experiences in the UK at the moment, lots of growers are worried about the alternatives for treating nematodes, such as stubby root nematodes or potato cyst nematodes. So with all of this in mind, us being here, us working on these alternatives is of the utmost importance. The good news is that there are plenty of alternatives to chemical control, which many members of this audience know well about. And these don't have to rely on varietal resistance or the standard cultural methods that we're all aware of. There has been interest in trap crops, for instance, such as being shown in this photograph. This is the species Solanum cimbreifolium, which is sticky nightshade and is used for trapping potato cyst nematodes. Can cause reductions of up to 75%. There are other trap crops, such as oilseed radish, that are used for trapping and reducing populations of beet cyst nematode. There are cover crops which are poor hosts or non-hosts for pests and pathogens. And there are those cover crops that are allelopathic. They are releasing compounds, metabolites from their roots that suppressed weeds or pathogens and pests in the soil. And this is an area that's been investigated over time, and there is a lot of potential in that area. There has been a lot of investigations undertaken on plant extracts, such as this neem plant here, and there are all sorts of plant extracts, including garlic. There is a whole other world looking at biopesticides, various microorganisms that can be used to treat pests and pathogens. But of course, we're here at this conference to learn about biocidal plants, and one of those biocidal plants is brassicas that are used in the process known as biofumigation. So being the first speaker of this conference, Vincent asked if I would give an introduction to biofumigation to those people who are less familiar with the process, so that you have an understanding of the concept and the variation in biofumigation. And if you study biofumigation and you look back over the references, you'll come across a very good statement or description that was put together by John Kierkegaard in 1998. And biofumigation simply means the suppression of soil-borne pests and pathogens by biocidal compounds, which are principally gases known as isothiocyanates, which are released in soils when glucosinolates in brassica crop residues are hydrolyzed. We'll talk more about that in the next slide. So this slide really is for those people who are less familiar with biofumigation, and this is the basic concept. So what we're doing in biofumigation is that we are growing brassica cover crops until the point that they reach flowering. We can see a crop here of Indian mustard, and these plants contain chemistry that's vital to the process, and we've got glucosinolates. These are secondary metabolites. There's 132 different types of glucosinolates that are known, and the profile of glucosinolates will vary in different brassicas. Some brassicas have limited profiles of glucosinolates, may only have 2, 3 different glucosinolates. Others have a more diverse profile of glucosinolates. Now, when these glucosinolates are broken down with a separate enzyme known as myrosinase, in the presence of water, we get biocidal compounds, which we'll come on to in a moment. But this is just to show you that in intact tissue, the glucosinolates are formed in sulfur-rich cells, and myrosinase is produced in separate cells known as myrosin cells. And this is basically an evolutionary defense mechanism against herbivorous insects, in that these compounds do not interact until the tissue is damaged, and then when that happens, then the biocidal compounds are released to deter the insect. So when we get glucosinolates and myrosinase mixing in the presence of water, we get hydrolysis, and to achieve this in field conditions, the traditional method is that once these plants reach early flowering, so between typically 25 and 50% flowering, then these plants would be chopped up as much as possible, bruised, chopped, to cause as much tissue disruption, and then those residues would be buried very quickly, as they are going to then release the gaseous compounds which we're coming onto. So what happens first is, during hydrolysis, glucose is split from the glucosinolate molecule to produce a transitionary compound known as an aglycone. We also get the production of hydrogen sulfate, and the unstable aglycone will then convert to a range of different volatile compounds. So we've got isothiocyanates, thiocyanates, oxazolidine thione, nitriles and epithionnitriles. So these are all products of hydrolysis. However, the most interesting compound in that list is the isothiocyanates, because these are the molecules with the most biocidal activity. Thiocyanates are of interest for weed control, because they're known to be suppressive to the germination of weed seeds. And these different compounds are affected by the soil pH. So the type of product that we get can be affected by pH. So if we have low pH, so below a pH of 5, we tend to get more nitrile production, and nitriles are less toxic. So that's something to keep in mind. If you're doing biofumigation, you do need a soil with a higher pH in order to get isothiocyanates, which is the key part of it. And biofumigation, as well as being produced by cover crops, you can also use preserved brassica materials, and I'm sure we'll hear about some of these, such as defatted seed mills. There's a lot of work in Italy through the CREA group. There is work that shows brassica hay can work in cut and carry systems. There are liquid formulations also come out through the CREA group, and in the US they've looked at powdered forms of brassica material. And whilst we're on this slide, we can see a nice cabbage white butterfly feeding on some of our brassicas there down in the left hand side. And you might say, well, that's a herbivorous insect, why is that not being affected by the biofumigation process? And the reason for that is that the brassica white produces a nitrile specifier protein, which means that during the breakdown of the glucosinolates, nitriles are produced rather than the isothiocyanates, and these are relatively less toxic to the butterfly and the caterpillars. Now, we talked about the main way in which biofumigation is done, and that includes growing a cover crop till the point of flowering and chopping and incorporating that tissue. However, something else that can happen is that if you grow brassica cover crops or any type of brassica, these brassicas naturally leach glucosinolates from their roots. And one thing I didn't mention is that the types of glucosinolates that you get in the roots and the shoots are quite different. And when these glucosinolates are leached into the soil, these can be broken down by a range of different microorganisms, and we can see at the bottom left hand side Aspergillus, but there are also bacteria that can break down glucosinolates, and this is because these microorganisms possess myrosinase. Now, as you would imagine, the amount of glucosinolates that are released into the soil will be at a low concentration, but during the growing period there is a long exposure period of anything that's within that root zone to that gradual breakdown of the glucosinolates into isothiocyanates. So it is possible that we can see reductions before we've even chopped and macerated the material. Indeed, we've seen this in our work, and this has been recorded elsewhere. For example, in France, there was a study by Matisse where they observed before the brassicas had been chopped, or even when they removed the brassicas from the field before chopping, they would still get a suppressive effect of Rhizoctonia root rot associated with sugar beet. So of course there's the full biofumigation effect, which is the traditional process: chop, macerate the material, get the hydrolysis, particularly in moist soil, and that is the main way in which people understand biofumigation. But then there is also what is referred to as the post-incorporation effect, and John Kierkegaard has termed this in the past the organic effect, which is to do with the fact that if you're incorporating organic material, then you are effectively creating a substrate for microorganisms that may be suppressive to given pests and pathogens. But there are also other compounds that come from the breakdown of the brassica material after incorporation. So initially, yes, it's ITCs, but later on there can be other compounds, volatile compounds that appear, such as dimethyl disulfide, which was picked up in some work that one of my previous PhD students was doing. So in view of the variation that occurs in the biofumigation process, there are a number of different factors that have been learnt through experimentation over the years, and this summarizes some of those points. And what I would say is that we are still learning about biofumigation. There are still facets of which we can change, modify, could possibly push the system, use the system in a different way. But essentially the aim with biofumigation is to maximize our glucosinolate content. That is our chemistry that's going to be active. That's what's going to give our volatile compounds in the first place. Secondly, we want to maximize our biomass. Biomass is what gives us all of the fragments of material that's going to deliver the biocidal compounds. So the more pieces we've got, in theory, the better biofumigation we should get too. And in terms of how much biomass should we be aiming for, we would be looking for trying to achieve a fresh weight of in excess of 50 tons per hectare if possible, a dry weight going up towards 10 tons per hectare, but typically dry weights may be much lower than that. The choice of biofumigant is also important. There are lots of different brassica species out there. As I mentioned, different species have different profiles, different concentrations of glucosinolates, and some of these may not be suitable for particular pests and pathogens. And it's very easy to generalize about total glucosinolate content and brassicas in general being effective as biofumigants, but there is some specific interactions between the types of glucosinolates and their isothiocyanates and given pathogens. Equally, the cultivar of the species is important. There are varieties that have been bred for biofumigation, and I would always refer back to the ISCI varieties, Caliente mustard, and varieties that have been bred by other breeders in Germany and the Netherlands, who have worked to produce varieties with high glucosinolate content. Also, the crop management is important. The selection of the site, the pH of the soil, too low, below 5 is obviously going to be a problem because of the selection of nitriles. Too much organic matter may lead to absorption of the isothiocyanates. The sowing date of the biofumigant is key too, in that if we sow too late and we're going into the winter or autumn period, then our biofumigant is not going to achieve the biomass that it requires. Equally, glucosinolate concentration is linked to the amount of UV, the day length, and the temperature. When we're starting to go into the winter, we're not getting those conditions. The nutrients, of course, is important for the crop. Nitrogen for biomass, and Glucosinolates are sulfur-based molecules, so sulfur is also important, and I'll show you some data on that in a moment. The cutting time is key in that glucosinolates, and we can generalize about this, but in general, the glucosinolates are reaching their highest point when we get close to flowering. There are some variations with different brassica plants, and there is some work that has been done on this. The selection of implements for cutting and incorporation, and I'll show you some work on this in a little bit, but essentially we want to use implements that create as much damage as possible, creating very fine particulate matter from that brassica material. We don't want lumps of brassica that's not been damaged. And the way that we incorporate it too, the depth of incorporation, a deeper incorporation is going to mean more mixing of the material throughout the soil. We don't want to be using implements that just bury the residues in a fashion that's not been mixed, like for instance using a plough. And having moisture incorporation, having a soil moisture that's ideally around 75% fill capacity is going to provide water for hydrolysis, but also the water within the plants as well is key for that hydrolysis to take place. This slide highlights how quickly isothiocyanates can be lost after the maceration of the leafy tissues of biofumigants, in this case Indian mustard. These measurements were undertaken in a field trial by William Watts using these devices known as VOC moles. We tracked the volatiles on Tenax, and the Tenax is sampled using GCMS. And what we can see here is that at 5 hours after incorporation, we can detect isothiocyanates, allyl isothiocyanates in this case, but at 10 hours, there is very little allyl isothiocyanate to detect. However, we can see that there is a low concentration of the volatile dimethyl disulfide that appears later in this particular assessment. Another point is that biofumigants are not suitable for every type of field situation. In the UK, we have cold weather during the winter, and frost or lying snow that freezes is very damaging to any late crops. And in this case, on the left-hand side, we see what happens when a crop of Indian mustard is overwintered. It simply doesn't work, and the accumulation of glucosinolates anyway would be low, as would be the biomass. Other things that we need to be mindful of is clubroot. Clubroot affects a range of different brassicas, not oilseed radish, and in the work that we did looking at clubroot some years ago, we showed that biofumigation does not reduce the viability of the resting spores of clubroot. Also, on the right hand side, we can see how some of the biofumigants are very susceptible to cabbage root fly. This was seen on white mustard and also oilseed radish in previous field trials. And cabbage root fly in the UK is a big problem on our brassica crops. So where we have crops like oilseed rape in the rotation, it may not be advisable to be practicing biofumigation. I now want to move on to some of our biofumigation case studies that we've done at Harper Adams University. So the first study I want to highlight is our work with Fusarium graminearum, and this is a PhD project that's being worked on by a student known as Samina Ashik, and this is a project associated with an EU project called My Toolbox, which is looking at different ways in which to manage mycotoxins. And in this study, what we were looking at is, could we reduce the viability of inoculum surviving on trash between winter cereals and maize, and principally looking at wheat. Fusarium is important for wheat and for maize. For wheat, it can cause protein loss, it can cause yield loss of up to 50%, and of course Fusarium graminearum is an important producer of the mycotoxins deoxynivalenol and zearalenone. On maize, we can get the diseases ear rot and stalk rot, and again we can have problems with contamination, and we can see a very heavily infected cob here on the right hand side. So to start in Samina's project, the first thing we wanted to do was to screen some isothiocyanates in order to select glucosinolates that produce those isothiocyanates in order to select the right brassicas for biofumigation. In general, what we saw was that Fusarium graminearum isolates were pretty sensitive to most of the ITCs other than ethyl ITC, and particularly to phenyl ethyl and benzyl ITC. And just to show this visually, we can see the effect on the radial growth here with the different doses of allyl ITC on PDA. As well as measuring the effect of isothiocyanates on the radial growth of Fusarium graminearum, we also wanted to see what effect the isothiocyanates had on the germination of the conidia. These asexual spores are important for initiating infections and also for spread of the pathogen. Here we can see the ED50s for both mycelial growth and conidial germination. Interestingly, if we were to just look at mycelial growth, we could say that 2-phenyl ethyl and benzyl would be the candidates. When we look at conidial germination, however, we can see that actually 2-phenyl ethyl and benzyl are quite poor at inhibiting conidial germination. However, allyl works on both mycelium and conidia germination. Hence why it's important to conduct these studies on both mycelium and conidia. So I want to show you a little bit more data from Samina's work. This is data relating to microcosm experiments. The microcosms are quite neat. They are simply jars or canisters that we fill with the same amount of soil, and we can amend that soil with the biomass that we would expect to get at an optimal field performance of biofumigants. And within those jars, after the fresh material is incorporated and water is added at the appropriate rate, we can add a sachet with pre-inoculated blind oat grains, and then this is left for a period of 6 weeks, and then we remove those sachets and isolate the oat grains to work out the incidence of germination. So these are the results from the microcosm experiment. We can see that all of the different biofumigants that were tested gave a similar level of reduction on the incidence of the germination of Fusarium graminearum from the infected grain. All of them decreased the germination of Fusarium graminearum. May be surprising considering our earlier work on the screening of the isothiocyanates. However, I would add that there are other volatiles other than isothiocyanates that are produced from the biofumigant material. So it is possible that there is an effect coming from other sources. Equally, brassicas have an array of different biocidal compounds. So looking at isothiocyanates is great. However, there is more complexity to biofumigants. I now want to share with you some results from our work with potato cyst nematodes. So in the UK, potato cyst nematodes are one of the biggest problems of potatoes. They cause losses of around £26 million each year, and if untreated, they could cause 80% yield loss. And this is a typical example of the elliptical patch that could occur in a potato field where there is a higher population of these nematodes. And the other problem is that PCN are very persistent, surviving for more than 20 years in the soil. So I'm going to show you some of our initial work, and you may have seen these before at the previous biofumigation conference, but I want to highlight them so that I can tell you the story. And this work was done by Bruno Engala, who's now based in France. And what we can see here is the effect of different biofumigant cover crops on PCN populations over time. We've got 3 different species, and we're measuring the viability of Globidira pallida eggs per gram of soil. The one thing I really want to draw your attention to, if I just put these up now, is the pre-incorporation effect. This was really surprising to us because obviously we expected most of the effect post-incorporation, but we can see that even before the brassicas have been incorporated, there is a reduction in the viable Globidira pallida eggs per gram of soil. So we went on to investigate the effect of this partial biofumigation seen in the field experiments undertaken by Bruno. And this is some glasshouse work, and it is a crude summary of the main results. So if you're interested in the detail of this, do have a look at his paper from 2015. On the left-hand side, what we can see is the effect of spiking soil where we've had biofumigants in with Synegrin, and then trying to recover it, and then looking at the microbial activity of that field soil. And what we can see is the more microbial activity, as identified by a fluorescein assay, the less retention of Synegrin, meaning that the Synegrin is being broken down by microbes in the soil and is being lost as isothiocyanates. So if we look on the right hand side, we can see that looking at that microbial enzyme activity and comparing it to the mortality of Globidira pallida eggs per cyst, we can see that the more microbial enzyme activity, the greater the mortality within the cyst. And this is because as we get more microbial enzyme activity, we're getting greater breakdown of Synegrin by those microbes, which are then producing isothiocyanates, which causes suppression of the cysts. And I think this is a really unique idea or observation, and one that could be taken a lot further in biofumigation. So these slides that follow show us how changes in our management plans can affect the type of biofumigation that we get. And this is some work that was done by William Watts as part of his PhD, and it really does show how well different incorporation treatments affect the biofumigation process. We can see on the left-hand side the axis showing PCN viability, and we can see how different combinations of maceration and also incorporation can have an impact on the suppression of the pest. And we can see in his particular experiment here, which is a combination of two experiments actually, that the spader combined with the flail mower gave the best reduction in PCN egg viability, highlighting that deeper mixing, better bruising and chopping gave a better effect on the PCN overall. Similarly, this slide, which was produced during some AHDB research that I was involved with, shows how important nitrogen and sulfur are for getting ultimately a better effect on PCN suppression. And we can see how nitrogen has an impact on the biomass, not surprisingly, but sulfur is having an impact on the glucosinolate content. And we can see that there is a limitation there. So if we look at the third set of bars in the top graph on the left hand side, we can see how add an additional 25 kilograms per hectare plus 100 kilograms per hectare of nitrogen, if we look now on the right hand side, gave us actually our best suppression of PCN. So that is almost like our recommendation, that we recommend 100 kilograms per hectare of nitrogen and 25 kilograms per hectare of sulfur. If you're interested to learn a bit more about our work on PCN and biofumigation, have a look at this pamphlet that William Watts and myself put together for the AHDB for growers in order to make agronomic decisions about biofumigation to get the best possible suppression of PCN. An important weed problem in the UK is blackgrass. Serious cases of this grass weed, which is extremely competitive when it has high populations, can cause yield reductions that are greater than 70%. And we had the pleasure of hosting Roberto Matteo from CREA in Italy to do a small project with us on blackgrass, and this paper was published in 2018. So initially, Roberto undertook some in vitro work to look at the effectiveness of 5 seed meals against the germination of blackgrass seeds. And it was very clear, very profound, that Brassica nigra caused 100% inhibition. So we took this further forward into a glasshouse experiment. So the results of the glasshouse study that was undertaken by Roberto clearly highlighted that different Brassica nigra doses of the dry seed meal, as they increased, we got more inhibition, and the greatest dose caused 100% inhibition of the blackgrass. In addition, in the study, Roberto looked at crude glycerin, as this is believed to be an underestimated co-product derived from the biodiesel chain. And in previous work that was done at CREA, it had shown good synergy with the DSMs. However, this was not shown in this particular study. Clearly, all of these studies highlight that whilst biofumigation generally has an biocidal effect, there is differences between their sensitivity, or the sensitivity of weeds, pests and diseases, to different biofumigants. So it is important to carry out these individual investigations. So in summary, approaches to crop protection are certainly in transition. We are losing lots of different actives all the time, and there is a greater movement towards environmentally sustainable farming. And this presents a great opportunity for biofumigation. Biofumigation can certainly be used as part of an IPM programme. However, as we've seen in this presentation, there are a variety of different factors that need to be considered in order to deal with the variation. And if we can understand all of that, we can optimise and improve what we're doing with biofumigation. The biocidal nature of biofumigants, as we've seen, makes them suitable for many different pests, pathogens and weeds. However, we need to avoid assumptions in that the activity of the biofumigants may be different for a different pathogen, a different pest and a different weed. And finally, we have a huge amount of work that's been done on biofumigation, a great amount of understanding, but there are still other aspects of biofumigation that need investigation, such as pushing what we can do with partial biofumigation, what can be done with regards to the after effects of biofumigation, and what other effects come out of brassicas. And there is some novel work going on at the moment looking at the blending of biofumigants with other species and how we might find synergies. I would like to thank people on this list, so our funders, the PhD students that have contributed to our projects, local growers, our trials team, John Reed and Tom Pope for supplying pest and weed photographs, and thank you to Vincent Mitchell for organising this symposium, particularly in the event of the pandemic and the changes that he's had to make, and thank you to the conference organisers for their kind invitation, and thank you to you for listening to me.